Hello, and welcome to the Logos Research Associates live stream broadcast. I'm Dr. Tim Clary, Research Director at the Institute for Creation Research. It's my privilege to be your host for this presentation. Logos Research Associates is a fellowship of scholars and scientists that are committed to upholding a high view of science and a high view of Scripture. We sincerely believe that scholarship, logic, and the scientific method can and should be used to show that the Bible's historical claims are both credible and ultimately superior to the secular model of origins. The goal of this live stream series is to provide you the opportunity to hear from top researchers who will convey cutting-edge scientific evidence for the creation account. You're going to be privileged tonight to hear another one of those researchers. But before I introduce our speaker to begin our presentation, we'll open in a short prayer. If you'd please join me in a prayer. Dear Heavenly Father, please be with our speaker tonight, giving him clarity, giving him the words to say, to convey his thoughts, and to get his ideas across to the people that are listening. And may his presentation touch the minds and the hearts of all who view it. We ask all these things in Jesus' name. Amen. I now have the distinct pleasure of introducing our speaker, and he happens to be my boss at the Institute for Creation Research, Dr. Randy Guliuzza. A little bit about Dr. Guliuzza. He's the president of the Institute for Creation Research currently. He's a leading biological design theorist. He's actively expanded ICR's scientific research and a critical endeavor to develop a theory of biological design, which he'll be talking about tonight. He holds a Doctor of Medicine from the University of Minnesota and a Master of Public Health from Harvard University. He served as Chief of Aerospace Medicine for the 20th Bomb Wing in the Air Force. Prior to that, he worked 9 years in the Navy in the Civil Engineering Corps as a registered professional engineer. Dr. Guliuzza also has a degree in theology from Moody Bible Institute. He is the author of multiple books and technical papers. At this time, I'd like to welcome, with the pleasure of extending a welcome to our speaker, Dr. Randy Guliuzza.
Well, thank you very, very much. It's a pleasure to be here, and I'm so glad to be able to speak to all those at Logos Research Associates. Thank you for the invitation, and thank you, Paul, for announcing this to a larger group of people. As you see by tonight's topic, it's a brand new thing, theory of biological design. In fact, it's an organism-focused theory of biological design that you'll see is really important, that phrase that it's focused on these organisms here. But I have to apologize. We have to get through a lot of material right up front, and we need to basically get it down in one big gulp, because you'll see one thing builds on top of another. We have to go through evolutionary theory, you have to go through the new theory of biological design, new models, and a head-to-head test. And most everything I'm going to talk about tonight has actually been covered in a paper that we published for the ICC earlier in last July. So if you want to review some of this in far more detail, I would encourage you to go ahead and download that paper, and it will tell you in exquisite detail a lot of things that I'll be talking about tonight. The main question is, why is this so important? Why should we even be thinking about a theory and focusing on theory? And one of the main reasons is because evolutionary theory itself is almost in crisis, not from the type that we were thinking back from the mid-1980s when Michael Denton wrote about it, but because a lot of evidence is coming in, a lot of observations of biology are coming in which do not fit the theory. And therefore, evolutionists themselves are divided over it, whether it really needs to be remodeled or redeveloped, or whether a third way needs to be developed. And as the little quote from that comment from Nature says on the side, this isn't really just a minor struggle. It's a struggle for the very soul of the discipline and for the very fundamental causes of evolutionary change themselves. And so what we find ourselves in as creationists or ID theorists is probably a once-in-a-lifetime opportunity where a lot of observations are not fitting theory. Theory needs to be redeveloped. The evolutionists themselves are in a conflict as to how that model needs to be developed. And we have a chance to actually frame the issue for ourselves for the first time going forward. We don't get this opportunity very often. And that's why we really need to be thinking about a replacement theory, not just to tweak the evolutionary change, but a complete replacement theory. Time to strike is now while it's hot. I guarantee you, in 5 to 10 years, the evolutionists will have circled the wagons, regrouped, and come up with a new model. And we run the risk of just copying that. So now is the time to be thinking about a whole new theory. So as I'm talking about theory, I really actually owe a little bit of debt of gratitude, believe it or not, to Stephen Jay Gould for his wonderful book, Structure of Evolutionary Theory. I really haven't read any book quite like it, which explains what theory is and what theory is supposed to do for science. But more importantly, it explains why Darwin developed his theory, how he developed his theory, how he studied actually a theory of design, which was prevalent prior to his own book. And Gould points out very, very carefully how he actually inverted many of the causations, and he completely turned most of the theory of design explanations on their head. So he studied a theory of design prior to developing his theory, and it helped as a framework to developing his framework. Well, since his framework was the anti-design theory, the future is actually back for us. And what we need to develop is the anti-design theory. And that will help us. So we need to actually understand the theory of evolution a little more deeply. And Stephen Jay Gould did a better job than anybody in explaining that. And basically, he says that theory rules everything. Theory sets up our research agendas, and it sets the questions we're going to ask along those lines. So what am I talking about when I'm talking about theory? As I said, I borrowed a lot of this from Gould's book, and I tried to summarize it into basically one sentence. And a theory is a working hypothesis, and there's 5 questions below which explain why it's working. But it tries to associate a lot of diverse observations that you see in a particular field. And you try to pull all those observations together into a testable idea that explains how something works. And sometimes it might explain how it originated, but more often than not, it's explaining how it would work. And so it's really not an inference. It is a complete framework. And the framework is intended to do 2 very big things. First, it's intended to answer the big questions in the field. So if we look at cosmology, obviously, Big Bang is the working hypothesis that's intended to answer a lot of questions in that field. And at the same time, it shapes interpretations of those observations. And that's why those 2 are bolded, because those are the main things that a theory is supposed to do. But in addition to that, as we look at observations, the theory guides in our question development. And in fact, it even sets the agenda of what questions are allowed and what are disallowed. And based on the questions and our previous interpretations, it helps us make predictions of what we expect to find. And therefore, all of these things taken together helps guide a research agenda. So really, it's the history of ideas which sets the framework. And that framework really has to carry 2 big loads: to answer big questions and to interpret observations. So when it comes to biology, there are at least 3 big questions. And obviously, this question number 1 is the biggest. How in the world did life ever get going on Earth? How did it emerge on Earth? And that has really a corollary question to it, which is really important. In other words, can natural interactions of inanimate building blocks, and those are biological building blocks, are those natural interactions sufficient to bring about life? Now, that's the big question, but actually, we're not even going to address this tonight. This is a completely different question, but we will address the next 2 big questions. And this is the one that has been stumping people for hundreds of years, and that is design or purpose. And speaking as an engineer myself, I know that purpose and design are inextricably linked together. You never design anything for a purpose, and the purpose always constrains the design. So the question is, how in the world do we account for the apparent design of organisms, which we see by that overwhelming appearance of design, their purposeful behavior, their exquisite fit to their environment, and the things that we see on those organisms? And these are questions which are actually, the ones in italics, are raised by evolutionists themselves. So how do we account for this apparent design of organisms? Or the corollary question would be this: is there an unguided natural mechanism sufficient to bring about the design of organisms? And Darwin supposedly answered that question. And then finally, number 3 is agency. In other words, why in the world do living things express what can only be summarized as a desire for things that it wants to do? And we call that agency, the ability to pretty much operate independently as an autonomous agent. And the corollary question to that is, can unintelligent nature actually function as an agent, which is sufficient unto itself, or is an intelligent agent needed to confer agency? In other words, do you need agency to get agency in these organisms? Now you can see there's a really big questions. And evolutionary theories purports to have the answers to these. And so theory is a working hypothesis, but in another way, theory is essentially a long narrative. It's a long narrative that is taught to people. And what it really does, in a way, is it kind of like puts little cubby holes in their mind where people will interpret things based on the labels that have been given to them for those cubby holes. And the cubby holes are arranged in a way which will explain the narrative. And these narratives are intended to steer people into a particular way of making sense of the observations in nature. In other words, they are meant to lead you to a conclusion. And that's exactly what Darwin's narrative does. And we also see, as people observe things and they interpret them, they actually put a narrative gloss on what they're seeing. So theories always have a goal in mind, and that is to get you to think in one particular way. And the way they do that is by putting these labels on cubby holes so that when you see something, you automatically want to store that in one of those cubby holes. And it doesn't really matter that all the cubby holes are filled. It's the way they're arranged, and it's the narrative itself which is actually doing all the heavy lifting. So tonight we're going to actually engage in three things to help guide our way. First is a case study. So as we go through this talk, I want to talk about cavefish. And we're all familiar with this. This is a major icon of evolutionary thinking. And I have two pictures of different types. You have a sided riverfish, which can actually mate with that hypopigmented or pinkish blind cavefish, and they can have sided offspring. But how do you go from a sided pigmented fish to a hypopigmented blind fish? And evolutionists think they've cracked the nut on that. And so we need to study this because I think it'll provide an excellent case study. Next, we need to contemplate two really important characteristics of organisms, which any theory of biological design or any evolutionary theory must address. In fact, a book was just recently published this year by Philip Ball, How Life Works, and it was reviewed by Dennis Noble, a physiologist out of England, and he summarizes it perfectly. He mentions that Ball grapples with the philosophical question of what makes an organism alive. Agency, that is the ability of an organism to bring about change to itself or its environment to achieve a goal. And he goes on to show and point out that Ball is not alone in calling for a drastic rethink of how scientists discuss biology, and that all of these people who are in this current struggle over evolutionary theory are arguing that agency and purpose, the two big questions that we just discussed, are really definitive characteristics of life that have actually been overlooked in current evolutionary theory, which is so gene-centric. And so we are actually at the heart of a major tussle of how we want to frame things and how we want to explain how organisms change. And then finally, we want to have a head-to-head test at the end of this talk that's going to test assumptions, the assumptions of my theory of biological design and the assumptions of current evolutionary theory. And I really even shouldn't say my theory of biological design. I'm going to talk about the theory that I'm presenting in this talk here, but others have been working on this for quite some time as well. And head-to-head test of assumptions was actually a major goal and purpose of experimentation. It wasn't just to find a mechanism or to identify a working part. It was actually to test whether our assumptions in our theories were scientifically valid or whether they needed to be revised. So as we go through this talk, hopefully we're going to understand engineered creatures in 4 steps. And the first step is actually the longest one. It's going to take about 30% of this talk here right now. And that is, we need to understand. Step number 1 is understand. Understand why current theory, current evolutionary theory, is constructed the way it is, or how it was constructed over time, as I mentioned earlier, as an anti-design narrative. And as we go through this, I hope we'll get a better understanding of evolutionary theory itself, but that will also help us build a better theory of biological design. So we need to understand that. And that takes us back then to the big question. How do we account for the apparent design of organisms? Or in other words, just put it in the pure vernacular, is it we have natural processes to explain it versus an intelligent agent, which we normally refer to as God. Which one of these is the explanation for this apparent design of organisms? And Francis Ayala, in his PNAS paper, probably his best PNAS paper that he ever wrote, believes that he has the answer, and that was Darwin's greatest discovery, which was design without a designer. And he's absolutely right. This was Darwin's goal. It wasn't to explain the diversity of life. It wasn't even to explain how life changed. Those were actually ancillary products. What Darwin was after all along was to explain this design without a designer. And for Darwin, this is what he said right here. With Darwin's discovery of natural selection, as we'll see here, he really didn't discover it. He invented a concept, which he called natural selection. The origin of adaptations in organisms were brought into the realm of science. The adaptive features of organisms can now be explained like the phenomenon of the inanimate world as a result of natural processes without recourse to a designer. So Darwin is focusing in right on that big question, which his theory is purported to carry. And he goes on to say this: Darwin's theory of natural selection accounts for the design of organisms and for their wondrous diversity as the result of natural processes, the gradual accumulations of spontaneous arisen variations, that is, mutations sorted out by natural selection. And he summarizes it perfectly there. Random, gradual mutations, which have been sorted out by, and that phrase right there, sorted out by, is going to become very important as it figures into Darwin's explanation and Darwin's theory. And so by this, in the end, he basically says that this was Darwin's fundamental discovery, that there is a process that is creative, although not conscious. Now, we should be initially thinking this doesn't make any sense, because nobody has ever seen a creative process that is unconscious or conscious in no way at all. But this is what he has sold, and this was what his theory purports to do. And Darwin changed the way, because of this, the way people thought about God. And he didn't do it, and this is what's quite remarkable, he didn't do it by changing how people perceived God himself. Darwin changed the way people understood God by changing the way they perceived his creatures. And that was really, really clever, because how you see the creatures is going to change the way you perceive the creator. And so we need to actually change our view of creatures back to what it was prior to Darwin. And Darwin did this through a very interesting way, and that's primarily through characterizations. And the words that are used by Darwin and the words that are used in evolutionary papers that are characterizing the object, and in this particular case, the creatures, are indicators of their workmanship, and these words convey ideas of origins. So, in many papers today, and definitely in papers prior to Darwin, and definitely in papers by ID advocates very, very broadly, the top design words that you would find in these papers, and I've read through many of them, I've pulled them out. So if we were to play the little game on this, the top words that we would find are these: purposeful, that's a great word; regulated, complex, targeted, orderly, specified, fitting and suitable, and precise. And these words are conveying something about the organism, but at the same time, they constrain or they control the way you think about the person who made those organisms. In fact, when you see those words, you're thinking of intelligence, and probably you're thinking of an intelligent engineer, because this is the way that engineers build things. That's the way that the computer that you're watching me is built right now. But Darwin changed the way organisms are characterized and the words that are being used in papers today. And those words are, as you'll see, the antonyms. In fact, those words, the primary word is purposeless. And then the next is random. In fact, those two are neck and neck with each other. On top of that is accidental, trial and error, broken, unplanned, cobbled together, messy. Now, when you're thinking about those words, It's also conveying some ideas about how they came about. And Darwin and those who have followed on have put these into their framework on purpose. They're purposely chosen to convey the idea of purposelessness or randomness. In fact, engineers really don't do things randomly, and they don't design in a purposeless fashion. So these words that are built into their explanations have an inherent anti-design bias in them. And just like so much of Darwinian vocabulary is built to convey the ideas right in the vocabulary, the explanation itself that Darwin put together is inherently anti-design. And Darwin focused in primarily on the one function of biological creatures, which is their ability to adapt. He could have focused on growth or reproduction or any of those things. But because organisms can change and they're not static, therefore, he can surmise that if they can change a little, that maybe over a long period of time and through the actions of his natural selection, they can change an incredible amount. And so therefore, whoever is controlling this explanation of adaptation is in many ways controlling the narrative. And this is why this theory of biological design is so important, because it will enable us to build models of adaptation, which are design-based and focused on the organisms themselves. So when Darwin was focusing on his theory of adaptation, which was meant to explain why organisms were designed, but really not designed, he's actually going to attack this in three ways. Darwin's anti-designer approach is going to interpret how organisms adapt, first, by replacing the engineering reality of purpose-driven and targeted solutions with random hit and miss outcomes. Now, there's a huge body of literature on this, and I've written multiple articles that you can find at icr.org on engineered adaptability, and some more recent ones as well, which point out through multiple sources why randomness is such an important concept in evolutionary thinking as a pushback to purposeful, intelligently designed organisms. Because no engineer would ever do things in a random, hit and miss, untargeted way. So when it's described in that way by the theory, it's already carrying the weight of an anti-design argument. And just to back it up, I'll just have one quote right here from Graves in 2012, which he summarizes classical evolutionary theories. He says, a classical or Darwinian evolutionary system embodies a basic principle, and that is purposeless genetic variation, purposeless genetic variation. And now that we've brought these words up, you'll probably key on them more, of reproductive individuals united by common descent coupled with natural selection of those individuals that fortuitously express the traits. It's a process replete with chance. And chance and randomness figures heavily on purpose in evolutionary theory because of its anti-design implications. Next, Darwin inverts the cause of organism change from internal to the organism, to external. And engineered things are always internally built. And here's just a couple quotes on this. And we go back to Stephen Jay Gould's Structure of Evolutionary Theory, where he points out that Darwin came up with an externalistic framework to interpret the cause of adaptation. And Gould says this: Darwin's theory, and these words are so important, in strong and revolutionary contrast, presents the first externalist account of evolution. Darwin overturned all previous traditions by thus granting the external environment a causal and controlling role in the direction of evolutionary change. And Gould's counterpart at Harvard, Rich Lewontin, added this: for Darwin, the external world, the environment acting on organisms, was the cause of form of organisms. The environment, the external world with its autonomous properties, was the subject, and the organism was again the object acted upon. And Kirshner and Gerhardt in The Plausibility of Life go a little bit further. When they talk about Darwin, that's the he. Darwin accepted the view that the environment directly instructs the organism how to vary. The organism was like modeling clay. And so therefore, we don't have active problem-solving organisms as any designed thing would be, internalistically controlled. We actually have passive organisms that are worked on by their environment and are shaped like modeling clay. Now, all these things are very, very important to understand evolutionary theory. And Gould goes back to point out, because it has to be random and non-purposeful, therefore variability and evolutionary change, he says, in short, must be copious, small in extent, and therefore undirected. And he adds that, in a sense, the specter of directed variability threatens Darwinism. Directed means adaptive pressures that automatically trigger heritable variation in favored directions. And he said that Darwin clearly understood the threat of directed variability to his cardinal postulate of creativity for natural selection. So that is how Darwin dealt with purpose, which brings us to the next big question, and that is this: how in the world did Darwin handle the need of an intelligent agent to confer agency? And he did that by, number 3, personifying nature as exercising pervasive agency to work on organisms as a substitute designing agent. And therefore, Darwin really added a vital element of agency to his framework, which I didn't catch for a long time, and many haven't caught for a long time, where he projects onto nature intelligence and volition. And actually, I was seeing this, and when I was reading through William Dembski's book, The Design Revolution, on one page, it was this glorious find, which was an affirmation of exactly what I was thinking. He points out that before Darwin, the ability to choose, that is to select, was largely confined to designing intelligences, that is to conscious agents, real agents with real ability to choose, that could reflect deliberatively on the possible consequences of their choices. He adds that Darwin's claim to fame was to argue that natural forces lacking any purposiveness or provision of future possibilities likewise have the power to choose via natural selection. In ascribing the power to choose to unintelligent natural forces, Darwin perpetuated the greatest intellectual swindle in the history of ideas. Now, that's absolutely profound. He closes with the fact that nature has no power to choose. And I, for one, was someone who was swindled by that idea. And therefore, I would always attach a verb to whenever I would use the phrase natural selection. Now, all natural selection can do. Natural selection acts on. Natural selection favors. Natural selection works. And there was always this idea of having a verb there, because when I was attaching this intellectual capacity to it, I was giving it and personifying nature as if it was the substitute agent. And this was smuggled in. So Darwin really didn't get an agency out, i.e. God's agency out. He replaced it with a smuggled in agency. And that is why other evolutionists, really very, I would almost say purist evolutionists, have a major criticism of Darwin, and that is he only swapped out one mystical agent, one that you can't see, feel, or touch, that is God, for another one, another framework for interpreting things with no measurable or quantifiable selective pressures, no way to identify what is being selected for or against. No, nothing in nature equivalent to God's brain or even a human brain to apply the word selection to it. In other words, he was swindling people. And therefore, Stephen Talbot, a very thoughtful, one of those anti-conventional evolutionary thinkers, points out that evolutionary biologists routinely speak of natural selection as if it were an agent. Natural selection becomes rather like an occult power of the pre-scientific age. And way back in 1992, when the evolutionary historian writing in Keywords in Evolutionary Biology, defining the term natural selection, pointed out that many people had trouble with Darwin's term for decades. And one of the trouble is that Darwin liked the term natural selection because it could be used as a substantive, that is a concept, not a real tangible thing, a concept, which could govern a verb, that is act on, work on, favor. And by doing that, he adds that such uses appear to reify, even to deify, natural selection as one of those agents. And Darwin, responding to all of this, by 1868, just about less than 10 years after the publication of his book, was doing some backpedaling. And he pointed out the term, this is a quote right from Darwin himself, the term natural selection is in some respects a bad one, as it seems to imply conscious choice. But he says, but this will be disregarded after a little familiarity. Well, obviously it hasn't, because people are still rebelling against it to this day. And then he adds, I have also often personified the word nature, for I have found it difficult to avoid this ambiguity. Well, of course he is, because you have creatures which look like they were designed by an agent. They exercise agency. Where did that design and that agency come from? And that is, he really can't get away from agency. He smuggles it in through natural selection. And we could really do the whole talk on this, but we really don't have time. So if you're really interested in probably one of the most scholarly discussions of it, we've recorded 3 one-hour episodes where we go right to evolutionary literature with explanation and dozens of quotes from major evolutionary theorists who discuss the problems with natural selection and why it is this misleading, mystical mental concept that Darwin had to smuggle into his theory. But he did it primarily to deal with those 2 major rocks, which are purpose and agency. And so beginning with the materialistic worldview, you have these anti-design assumptions, which gives us, as I've already pointed out in the scientific literature, the relentless drumbeat that organisms are purposeless and change is accidental or random, which leads to broken and unplanned things. And all of those words are summarized in the word mutation, which I know has multiple connotations, but in the mind of most people, it carries the idea of a mistake or something that is broken, something that is completely purposeless, something which no engineer would ever do. And that is how it carries the water for anti-design thinking. And then on top of that, you have other anti-design assumptions that nature can act like a breeder, of which we get the relentless words in the scientific literature where nature acts on, nature favors, nature works on, or even as Jerry Coyne says, nature can see, select, save, and build, which is all summed up in the word selection, which gives you the mystical substitute agent. So in a quick summary, this is how evolutionary theory deals with 2 big rocks, 2 big questions. Why do organisms look designed? And can nature substitute as a sufficient agent? And they argue that it can. And this slide here summarizes how it did it. Now, if you were to turn to our paper, our ICC paper, I put in there a table. And this is the table here, and it summarizes all of the major points of evolutionary theory, what their assumptions are, what their basic premises are, and how they will interpret observations in a single table on that. So looking at it carefully, Darwinism is fundamentally externalism. It focuses on the environment as an active agent molding and shaping creatures. And it has some basic assumptions in which they empower nature to act like a human breeder. And therefore they confer onto nature. Actually, they see things which have never been quantified, but they call environmental stresses selective pressures, which seem to be able to drive creatures through lethal types of sieves. And based on their random thinking, they believe that nature is able to, through a tinkering or a cobbling process, cobble together in a very purposeless way highly designed organisms over a long period of time because they're acted on by this natural selective force. And in their view, natural selection becomes the overriding force driving evolutionary change. And as I mentioned, Gould already pointed out that there are 3 basic assumptions is that genetic change is fundamentally undirected. In other words, there's no automatic activation of change in any one direction in any way. There's no purposeful outcomes. They believe it has to be gradual. In other words, no wholesale arrival of new traits. Though that's changing because it's being challenged. But at the time it was put together, the wholesale arrival of traits, in their view, was tantamount to a miracle. And therefore, change had to be imperceptibly slow. And therefore, the genetic variety had to be very, very large and plentiful within a population. And therefore, selectionist thinking is primarily population-based. And with those basic premises and with these basic assumptions, they will interpret what they see around them. So when evolutionists, based on their theory, would see a genetic change because they assume that it is undirected, they don't have any tests to demonstrate that, they assume that these are genetic mistakes that offer the principal source of genetic variability. They assume that these are random. They believe that these will lead to a loss of function and broken products will be the dominant outcome. And they believe it's non-Lamarckian. And this explains why Lamarckianism was so vilified in evolutionary thinking, because Lamarckianism in their thinking had the idea that there was a goal or there was a progress or there could be purposeful inheritance of predictable traits. And they invented a concept, which for much of my thinking, I thought was a real thing, was the Weismann barrier, which there will be no transfer of genetic information from somatic cells to germ cells. This was all invented, and this is how they interpret things because evolutionary theory demanded it. And because they believe it's gradual, they will then interpret these changes as slow and linear and continuous over time. And of course, because they believe that that there has to be a copious amount of genetic variability in the population, their method of how these things are sorted out is through a complete trial and error or hit and miss process. In other words, you have a lot of traits within the population. Only a few of those will actually solve a problem. And it's through this trial and error process of which everything gets sorted out. So undirected, slow, trial and error, random, purposeless, all of these taken together are what give evolutionary thinking its strong anti-design appeal. So therefore, obviously, if we're going to build a theory of biological design, we need to address these major assumptions, because these assumptions with evolutionary theory are actually the opposite. They were built purposely to be the opposite of the major assumptions of a theory of biological design. So step number one was to understand. Step number two is to embrace a new approach to biological research that accepts the observable engineered workmanship of creatures. In other words, it accepts purpose and agency. So some may be thinking, well, why should we do this? Why can't we just modify evolutionary theory? Why can't we just show its limitations or show where it's essentially wrong? Is there really, really a need to move from Darwin to engineered biology, or as I say here, to move from Darwinism, a way of interpreting a worldview, back to engineered biology, which is how biology was viewed long before Darwin? I'd say there's a desperate need. There's a need only today because evolutionists are in turmoil, but because we've been getting explanations wrong ourselves. So going back to our case study, how in the world do we explain observations of these two fish that are the same species? One is sighted and pigment, and the other is blind and hypopigmented. How in the world do you get from that? Well, the typical answer that we're giving today, and I'm using the umbrella of ID very, very broadly, of which even creationists would fall under that umbrella, is this: that Darwin's mechanism is essentially true. His selectionist mechanism is essentially true, but it's limited. In other words, it can't really do everything that Darwin has claimed. And now, where would we find such an explanation like that? Well, let's go to an exhibit on cavefish. And let's read how this exhibit explains how these cavefish formed. And this is the typical explanation. As genetic information is copied and passed on generation after generation, occasionally there are copying mistakes known as mutations. And that's exactly how most people think of a mutation. It's a mistake. Mutations have been observed to destroy, damage, and corrupt genetic information or be neutral, but have never been observed to add new information. This is true even of so-called beneficial mutations that may be advantageous to the surviving organism in some circumstances. When a mutation occurs in a light environment that causes an animal's offspring not to have eyes, it is an enormous disadvantage. So natural selection, and here's the verb, they're always there within one or two words of selection. So natural selection eliminates this flaw as if it's a personified agent. When the eyeless defect occurs here, that is in a cave, it does not give any disadvantage, so it is not eliminated. In fact, it gives advantages. Those with eyes can crash into things, injuring the eyes, and can get diseases of the eyes, possibly leading to death. Eventually, selective pressures ensure that all are eyeless. These ghostly fish are prime examples of how mutation and natural selection can lead to a reduction of functioning eyes. These adaptations are no evidence at all for the belief that complexity has arisen by such a process. They only show how information can be lost in the world. And so where would you find such an explanation like this? Well, this was in our original museum, the Institute for Creation Research original museum, on how we explain the origin of cavefish eyes. And so summing this all up, basically what we were is theistic selectionists in explaining this. And so our explanation of how you went from a surface-dwelling ancestor to a blind, hypopigmented one is that these creatures gradually morphed into the cave-dwelling forms, that it was due to random genetic mutations which led to broken systems through a purposeless trial and error process that resulted in a loss of information that produced depigmentation and blindness, and selective pressures ensured that all are eyeless. And I would submit to you that this was not a formidable response to evolutionary explanations because it is fundamentally an evolutionary explanation, regardless of whether we added the caveat that all this shows us how to break something and not to make something. So this is not a good response against evolutionary thinking because it reaffirms all the major tenets of current Darwinian evolutionary theory. But it also has some problems, which leads to some major harms. And first, it derails scientific research into not looking for the non-random. And I know this is true because I read a lot of papers on these cavefish, and nobody is currently looking for non-random explanations. And I have to say, neither were we prior to this. It derails scientific research and not looking for the non-broken. Evolutionists are looking for broken systems. Mutations for adaptation, it's totally anti-design because no rational engineer in their right mind would ever use broken things, random broken mechanisms to ever build things. It leads to a theistic selectionist explanation because it's just really a lighter version of real evolution. And these are just four, and I could go on with many of the harms that are not only to theology, but also to our practice of science. So the question is, can we actually offer something better than to concede Darwin's framework and then to simply to argue that evolutionary explanations are inconsistent? In other words, it's irreducibly complex and you can't explain that. Or insufficient. Look how complex these things are, and they defy your evolutionary complex. Incomplete. Evolution doesn't explain, or inaccurate, or any of the other things that we're having problems with. Why just try to pinpoint the problems with this? Why not replace it altogether? And that's why I would keep on referring to this wonderful quote, which I wish I would have come up with, but was actually devised by Paul Nelson. And he summarized exactly what we need to do. We need to begin doing biology as if Darwin had not been born, and think about it in a completely different way, not modify it, not tweak it, but come up with a real theory, which is a real working hypothesis, which actually guides interpretations of our observations, helps us frame questions, and sets a real research agenda. That's what real theory will do, and not just pointing out that Darwinian evolution is inconsistent or incomplete or inaccurate or whatever. So if we're going to build a theory of biological design, we need to come up with some assumptions. If it's going to be engineering based, we need to look at how engineering and biology might actually relate to each other. And this picture kind of summarizes the current state of affairs, that engineering, well, man-made things are best explained by engineering principles, and workmanship explains those. Biology has life and consciousness and agency and the major biological functions of growth, reproduction, metabolism, and adaptation. And maybe engineering has something to explain about metabolism or adaptation, but it certainly can't explain life. It can't explain agency. And as far as we know, it can't explain reproduction or growth on that. And so we kind of separate these two into distinct realms. But I'm going to argue that there actually can be overlapped, and I'll show you how in just a few slides. But let's begin with the working hypotheses and some of those basic assumptions that we might have if we approach organisms as if they were really engineered. In other words, they look like they're engineered because they really were engineered. And hypothesis number one, or you might call it one of my basic assumptions, is that biological functions will be accurately explained by models developed utilizing engineering principles. And I could go through several of those, but I'm going to show one model later on that explains adaptation on that. But we could use it for growth. We could use it for even reproduction. We could use it for other things. And Winston Ewart, an intelligent design advocate, has come up with another model to explain the hierarchy that you might find in living creatures. It's called a dependency graph. It's also one that human engineers and other researchers have developed. Working hypothesis or assumption number two would be this: that analyzing human engineering practices will be useful for two ways. It'll inform biological research and predictions, and it can direct researchers to accurately characterize biological phenomena. In fact, I would say, unless they're really accurately characterized according to engineering principles, then you're probably going to be off the mark. And because there's going to be this overlap between engineering and biology, I really expect to find corresponding systems, in fact, and system elements between human engineered things and those which we find in biological systems performing very similar functions. So this is a prediction. This is a real theory. This is testable. I mean, I'm putting it out there that I'm expecting to find corresponding elements. You can look for these, you can test them, you can see if they're really there or not, and you can modify the system. A theory of biological design would also expect to find purposeful systems within and between all biological functions in organisms, since the engineering of complex systems is always top-down. Another working hypothesis is that basic research of biological functions and technical applications are both within the domain of engineering practice. In other words, biologists do both basic research and they do applications, and a lot of bioengineers do applications and also basic research. And I have a couple reasons why I might have this hypothesis. First, as far as I know, no biological function has yet eluded an explanation by engineering principles, and this would be good during the question and answer section. Next, biological systems can be reverse engineered. There is a known consistency between human engineered systems and biological systems, and it's actually very broad. Engineering principles are currently being applied in biology, and engineering principles are increasingly being recognized in biology. So in so many ways, bioengineering and biomedical engineering is the wave of the future in terms of biology. So everything is headed towards this way anyway. But I need to put a major caveat, and even evolutionists recognize this, is that there's two distinct biological categories between biological functions and the hard questions. And František Beluska raised this question and he summarized it very well by saying, however, in order to complete this revolution, which started with the discovery of DNA and these biological functions, something fundamental is still missing from modern biology, as evidenced by difficulties in tackling the hard questions. What kind of hard questions? Hard questions such as biological nature of feelings, emotions, and qualia, and consciousness. This is not any trivial problem. However, the true nature of these inherent phenomena is still elusive and highly controversial. And I would suggest the reason why why it's so elusive to explain life and consciousness and agency, is because these are immaterial things. They're not reducible to engineering principles, nor are they reducible strictly to biological functions. In other words, your thoughts are not just the product of the interactions of your neurons. And because they're immaterial, humans cannot create these. We can't create life. And this also explains why creatures are not mere machines. But in terms of growth, reproduction, adaptation, and so on, these can be explained by engineering principles. And therefore, engineering principles explain how any type of workmanship, man-made workmanship, or even God's workmanship, actually operates. And one fundamental assumption of that is anything that we create, such as humans building a satellite, or building a car, or building a computer, anything you build, you must put into it upfront its capabilities. If you don't design those capabilities into it, even the capability to change, even the capability of AI, or even the capability to relate to things around it, it will never get those capabilities. And so therefore we will move from an externalistic framework to an internalistic framework. And here's another important feature of our paper published at the ICC. And this is a table which summarizes in one table the major premises and assumptions of a theory of biological design. Now, this is obviously a work in progress and can be contributed to by many. But first and foremost, it is a system which is not externalistic. It is primarily internalistic. And it is an engineering-based framework, and it can be applied to biology. And therefore, as it says there, it's integrated, comprehensive, instructive, and directive. In other words, basic research will guide all of these things. Biological systems related to development and reproduction and all can be reverse engineered. That engineering will actually guide our research and directive in those areas. And therefore, contrary to what the evolutionary basic assumptions are, basic assumptions of a theory of biological design would be, there's three: intentionalistic. In other words, it's purposeful. I expect, and those who would develop a theory of biological design would expect goal-directed activity, and that goal direction to be organism-wide. It would be internalistic. What I mean by internalistic is that I would expect to find, and this is a test, identifiable control systems within the organism as the true cause of all of their operations. This is a prediction. This is a testable prediction. This is part of a real theory. And it would be individualistic. In other words, organisms are autonomous agents. They're delineated by a self boundary, which marks self from non-self. And we can't go through all of the ways that we would interpret things, but the first one is very important. Contrary to evolutionary theory, when they see a genetic change, they interpret it as random or broken. My default position, until seen otherwise, is I will interpret that genetic change as directed. It would be coherent. It would be foresighted and proactive. And you can either download this program or a paper, and you can see how those would be explained. But in other words, when I see a change, my default assumption is that it's a purposeful change, particularly when it's adaptive. I recognize there are genetic changes that really are copying errors and copying mistakes, which are overwhelmingly non-adaptive. But when I see an adaptive genetic change, my default initial interpretation will be that it's purposeful and that it's internalistic. In other words, these identifiable control systems will be within the organism. And therefore, as it says there, there'll be a precedence, which is the second one there, which is the organism's traits determine all of its capabilities. And therefore, whether an organism can solve an environmental challenge or not, if it does, it gets the credit. If it doesn't, it doesn't. But I'm not going to say that it was favored or is acted on. The precedence goes to the traits which can either solve problems or they're not. True triggers are always a part of the organism, just like in any design thing. And all true triggers, all true triggers are actually a sensor of one type. And then finally, it's going to be individualistic. which primarily says there's a boundary between self and non-self. And so if organisms are going to relate in any way to other organisms around them or to any other external conditions, they will have built-in interfaces which enable them to do that. And these interfaces will actually also have other identifiable and predictable elements, such as authentication, protocols, and a common medium. And organisms will define for themselves what external conditions are stimuli and what aren't stimuli. So you can see a theory of biological design has a lot of predictions, it has a lot of way to interpret what we see around us, and it is diametrically opposed. It's almost the exact opposite of what Darwin and those after him have come up with to explain adaptation and the like. As an anti-design explanation, this goes right back to basic engineering elements, lays out the basis for a theory of biological design with real premises, real assumptions, and real ways to interpret what's around us, frames questions, and can direct our research. And this is what constitutes a real scientific theory. And you must have these things as part of it. And so we've kind of gone over this. And as I mentioned, it's a work in progress, so it can be changed. And then finally, in the last 5 to maybe 10 minutes here, I want to go over step number 4, which is build new models. Now that we have a theory of biological design, and as you can see, we have to kind of like get this all in one big gulp, we can begin to build models. And as I mentioned, whoever controls the explanation of adaptation is kind of controlling the narrative. So let's work on a model which is going to explain how organisms might adapt, a model which can actually be tested and put to the test. And the model that we're going to build is going to be based on what we're actually reading in current scientific literature. And the words that are characterizing adaptation today are not random, broken, and the like, except in their explanations and conclusion sections. But when you read the actual findings, you find words characterizing adaptation as regulated. In fact, many papers classify it as highly regulated, not slow or gradual, but rapid, repeatable, sometimes even reversible, and with targeted responses which are so precise to the external condition or the problem that they're trying to solve that they're even predictable. Now, words like regulated, rapid, repeatable, and predictable are actually describing an engineered methods. So at ICR, we've developed a model for adaptation, which we call continuous environmental tracking. That's a descriptive model of what we say organisms are actually doing. In other words, what creatures seem to be doing is continuously tracking the environmental changes within them and around them, which then leads to purposeful adaptations. But how that actually happens is not through a mechanism, but through many mechanisms, actually dozens of different mechanisms, which enable these organisms to make their own self-adjustments to solve these different problems. And therefore, we're looking for characteristics of responses which would be similar to characteristics of what we would find in human designed systems. And so if humans have designed a tracking system, we expect to find sensors, we expect to find internal logic, and we expect to find an output response. And therefore, what would distinguish continuous environmental tracking from a Darwinian explanation? Well, first, as we've already pointed out, it's internalistic. In other words, CET is organism focused. And this makes sense. If you want to figure out how a space shuttle works, you look inside the space shuttle. If you want to figure out how these cavefish work, you have to look inside the cavefish. It will be focused on them, and all explanations must be focused on these organisms. Next, CET is an engineering-based explanation. As I mentioned, some of our basic assumptions is that engineering principles will be the best explanations. And in fact, from my own personal opinion, they will be the only correct explanations. They're only going to get you on the right path of research right from the beginning. And that engineered capability is always put in up front. Just like you put in the capability for a space shuttle to solve all different kinds of environmental challenges, the ability to solve those challenges precedes the challenge. The solutions precede the challenge. They're not due to the challenge. Randomly selected or favored, these solutions precede the challenges. And therefore, we are looking for corresponding system elements. And just as that car designed by GM, which is a fully autonomous vehicle with all kinds of sensors and computers on board with programmed logic, if it does this, then it wants to respond that way, and output responses, could drive autonomously if it had enough fuel from San Francisco to Miami, Florida, going around all different kinds of obstacles because it has sensors and logic and output responses. Organisms would do the same thing. And that's why I'm looking for corresponding elements when I read a headline like guinea pigs beat climate change by tweaking their own DNA. Now, the experiment that they're describing there is, is how they took 5 male guinea pigs and wanting to know how they respond to climate change by putting them in cages which are heated on the bottom. And they mated these guinea pigs after 70 days or one full cycle developed sperm with females which had not been heated. And they had offspring which had genetic changes which were already there in one generation, suitable for dealing with climate change or higher temperatures. And I would therefore look and predict to find a temperature sensor, an internal logic, and mechanisms to actually direct those changes. I'm looking for those same kind of corresponding systems, which is how we develop a research agenda. And I've mentioned this in prior papers, so I won't go over them in the past, about how organisms like the rove beetle can actually morph when it detects army ants into looking like an army ant and smelling like an army ant and behaving like an army ant, and not just one, but into 9 different species of army ants when it encounters them. And I also discussed how stick spiders that you find in Hawaii, which are either gold, like the one that's pictured there, living on tree leaves, or brown on tree trunks, or white living on lichen, can actually transfer from one island. Let's say a gold one goes to another Hawaiian island, and it can morph into the brown and white forms before those forms ever get from the other island. And how these researchers predict that the Aramna spiders have some sort of pre-programmed switch in their DNA that can be quickly turned on to allow them to evolve rapidly into these successive forms. But how that process might occur is still unclear. Well, I would have a process based on a theory of biological design, start looking for the sensors and the internal logic and the controls and looking for purposeful changes. I covered another paper how E. coli bacteria in separate populations exposed to the same challenging conditions all develop the same genetic changes. And therefore, our title was Predictable Evolution Trumps Randomness of Mutations. And here's another model, which just came out in 2022, is an incredible model, almost using the exact same language as the CET model. Direct observation of adaptive tracking on ecological timescales in these flies, of which they had 10 different populations of flies, which exploded to almost 100,000 flies per population. And they tracked them over 4 months, and they noticed that there was over 60% change in their genes, and tracking specific genes, which they thought were useful for fitness, were changing in lockstep over that period of time. So that the researchers from University of Pennsylvania describing this mentioned that in earlier studies at the Philadelphia orchards, Smith, who was the lead researcher on this, and colleagues had seen that fruit flies present at the beginning of the growing season, were quite different from those buzzing around in late fall in measures of stress tolerance, reproductive fitness, and even pigmentation. But that research couldn't rule out the possibility that new flies were entering the population and causing dramatic shifts. But with their research on these hundreds of thousands of flies that I mentioned, they pointed out that over the course of their experiment, which just lasted just 4 months, the research documented changes in 60% of the fly's genome, with this direct observation a swift and continuous adaptation in response to the environment, a phenomenon known as adaptive tracking. Biologists have established a new paradigm of how to think about this on a time scale. And what was more remarkable than anything else is that the population itself They saw was able to track differences in the environment, said the lead researcher. This was not a response to a single selective event, such as a drought. The populations were constantly evolving and shifting throughout the entire experiments. He suspects that other organisms can do this as well. And what made the findings particularly compelling was that the direction of adaptation changed multiple times, swinging like a pendulum as environmental conditions can change. Now that's phenomenal. And that's how we can build a new model which can actually explain this far, far better than random changes, but through purposeful changes. And that brings us finally to the last point, how to conduct new research which can put the theoretical assumptions of a theory of biological design or continuous environmental tracking, what those basic assumptions are, into a head-to-head test with Darwinian thinking, which brings us then back to the question, how in the world do we explain this change from sided and pigmented forms to the hypopigmented and blind forms? Well, ICR's Cavefish research tests the important assumptions of evolutionary theory compared to ours. And we would have these basic assumptions that organisms are changing based on sensors with built-in condition consequence logic, which would lead to suitable responses. And therefore, if they detect similar conditions, such as the stresses of a cave, and they have similar internal programming, which would make the changes that you see there on the screen, then we would expect similar responses, not only in blind cavefish, but also in blind salamanders, crayfish, insects, spiders, and on and on. We would expect to find these as probably purposefully directed changes. But evolutionists have a completely different explanation. So I'll read to you just 3 quotes from their papers on how they explain what they see in these fish. From Borowski, one of the leading evolutionary researchers, points out there's loss of function mutations in the oculocutaneous albinism gene, which have been characterized in 3 independently evolved populations. In other words, it happens over and over again. And he considers these mutations. And as he says, for reasons currently unknown, OCA2 seems to be evolution's favorite for pigmentation regression. So you see all of their assumptions, broken, random changing, nature personified as it's his favorite target, and something happening over and over again. Now, when we see that from a theory of biological design, we should be thinking there's a purposeful reason for that. Jeffrey, another leading researcher on cavefish, points out that mutations have occurred in both the Pacone and Molino OCA2 genes. The OCA2 gene is repeatedly targeted in cavefish, resulting in independent origins and albinism. And another leading researcher, Jeffrey and another one of his colleagues there, point out that there's another gene, the melanocortin-1 receptor gene, which has been mutated repeatedly to generate the same cave-like features in different cavefish populations. So summarizing, based on their theory, what they predict and how they interpret what's happening, they're interpreting these genetic changes as random broken genes, which overwhelmingly result in broken pathways for pigmentation, which would lead to a loss of function for that pigmentation, and they are definitely not expecting any specific consequences due to innate programming. But the CET prediction obviously would be very, very different. Based on engineering assumptions, we're expecting genetic changes which are purposeful. We're not expecting broken pathways. We're expecting down-regulated pathways, not broken pathways, down-regulated pathways that can be modulated up and down as needed. This allows us to put their observations, their interpretations, their predictions to a head-to-head test with what we would predict. And so we did some initial experiments, and this is a blind cavefish, obviously hypopigmented and blind. And we reintroduced this fish back into simulated sunlight conditions. Now, if it was a broken mechanism, this fish would stay pink and non-pigmented. If pigmentation is a purposely driven up and down regulated change, we would expect pigmentation to return. And after 32 days, this is the same fish. This fish is actually still alive. This is what we found. a fish with returned pigmentation. And that's the female, and this is the male of that fish. Both of them regain pigmentation. What a confirmation of a modulated system, completely contrary to those assumptions. So we repeated that experiment, and there you see a pigmented fish at the top and a hypopigmented one. We bred the first generation of those pigmented fish, and this is what we got in the successive generation. A hyperpigmented fish, which pigmented twice as much in half the time. Now, this is a confirmation that this pigmentation process is clearly not a broken system. They're probably not broken genes. It's something which can be regulated. And we can actually quantify this pigmentation in these fish right there. We can quantify where the pigmentation is happening and how much is happening. But what about the reverse direction? How can you go from a pigmented fish to a hypopigmented fish? Well, this paper by Rohner in 2013 indicated that the water conductivity or the degree of ionization of water might play a role, because they found that ionization regulated heat shock protein 90 could lead to hypopigmentation and a decrease of variability in eyes within a single generation. So that's exactly what we tried. We tried experimentation with fish being placed in deionized water. And so this is the same fish. There's the original surface fish with its pigmentation, and we placed it in deionized water. And after 2 weeks, as you can see, the pigmentation has essentially vanished. But it actually was happening much faster than that, because we repeated the experiment. And this is the same fish again. This is the surface fish, as you can see on the top with its pigmentation. And after being placed in the deionized water for only 2 hours, you can see the loss of pigmentation and the pink color shining through from underneath these. So CET has predictions. It is a real verifiable model, and it can actually be tested, where we predict that pathways can be either upregulated or downregulated. And we offer a testable model that can be placed head-to-head with them. And therefore, continuous environmental tracking is actually an anti-Darwinian explanation, which is thoroughly engineering-based, completely focused on organisms, like how you would design and build anything, purposeful, non-random mechanisms, without any mystical assumption or events. And then finally, the last slide, what can we offer in exchange for the random, adaptive, non-designed processes which evolutionists have, where they see organisms, and this is a quote, as passive organisms shaped by the vicissitudes of nature. Adaptation is characterized as undirected, trial and error, hit and miss, where changes be iterative, gradual, or linear, or as even Darwin said, numerous successive slight modifications. We can offer a fully designed and purposeful adaptive process, which focuses on the organisms and expecting to find adaptation characterized as regulated, with targeted solutions which are predictable and repeatable often, and many times reversible. And the variability is doesn't force to be characterized in any way. In other words, it can be rapid or slow if need be, episodic, go one way or go back, as we found with the fly experiments. And it is completely and functionally coherent. Now, I know that this was a long talk, and I know it was one big gulp at one time, but everything has to fit together. The understanding of the evolutionary mechanisms, why they were anti-design, and how we can go back to building a theory of biological design with true models which can make true predictions. So thank you very much for your time. I really appreciate that. And now I'll turn it back to Dr. Clary for question and answer.
Well, thank you for an incredible presentation, Dr. Guliuzza. We do have some questions that have come in, and there'll probably be some more as we open this up to more questions. It was a big gulp. I hope a lot of people can get that all in, almost like a cavefish gasping for air. So here we go. Let's see. We had a couple questions that will kind of go in the order they came in. There was one that was asked about about God's workmanship a little bit. Shouldn't there be a design argument for non-biological things? And he suggests that God surely made Niagara Falls, just as he made a fruit fly. So in scriptures, he talks about in weather, mountains, etc., described with design characteristic languages. So is there some sort of, design argument, I guess, for non-biological things?
Of course, yes, I would thoroughly agree with that. Many non-biological things. In fact, this is one of the major premises of even intelligent design theory is the fine tuning that we would find in the universe around us. And not getting into a theological explanation, in other words, just appealing to God's absolute sovereignty, but I see how we relate to the environment around us, how we relate to those conditions, how many of those conditions are absolutely essential for us to function. In other words, we're an autonomous entity with a distinct boundary, but that doesn't mean we're independent of various needs from the environment. We need nutrients. We need to get fuel, and how that fuel and how those things are put together and how we are able to turn those into building blocks for our bodies and for energy. Yes, I would say there's a strong case for design in non-biological things as well. And that's one of the major premises for fine tuning.
Well, in geology, we believe that God created the earth, spoke it into existence as a young earth creationist. And so there had to be design, obviously, into that as well, and including everything that's on the surface of the earth. That's a good answer. All right, here's another question. You did some of this, and you answered this towards the end of your talk, particularly, where this question is, do we make enough predictions? And it goes on to say, or maybe we don't clearly state our predictions. And then the guys, kind of a follow-up to that question is, has ICR published predictions that CET would make for the Cavefish research, which you just kind of did? So here's the kind of the question right there. Kind of two questions. Do we make enough predictions, first of all, as creation scientists?
Well, we haven't been making enough predictions, primarily because we don't have a model and we didn't have a framework in order to frame the questions and interpretations and make predictions of what we're looking for. But we do make predictions. In 2012, I put together an article, even in Acts and Facts, and made a prediction because I was seeing all of these organisms with not just the cavefish, but with all of these features which are custom fit for the caves, that it began to dawn on me, well, this is probably an engineered system which is designed not within fish, but within other organisms, enabling them to fit and fill caves. So in 2012, I made a prediction that we would find a design mechanism which could control eyes, in other words, eye regression and perhaps even eye redevelopment, control pigmentation, and many of the other traits that we would see to fish. And I published this in Acts and Facts with a prediction. And of course, in 2013, Roner's paper came out and we had the prediction. Obviously, tonight, I don't know how many I laid out, but I laid out at least half a dozen other predictions, which can be testable on this real theory of biological design. And even other creationists in the realm outside of biology made predictions. I know Dr. Russ Humphreys made predictions on what he expected to find in terms of magnetic fields and other things when spacecraft zoomed by those celestial bodies, and his predictions were right. But the test of a really good theory is how robust of a prediction it can make and if it actually stands the test of time. And so this is why it's so important to put together not just arguments against evolution and not just tweaking evolution, but to put together a real theory, as I try to outline in that table right there, with basic premises and assumptions and how we will interpret and what we expect to find, not only to make predictions to validate those, but also to guide our research projects.
No, the last part of that question was, has ICR published predictions on that? And you have published at the ICC a big paper on the cavefish this last summer. So they can go to the proceedings of I believe it's the 9th ICC. You can go online, look that up, and look up the proceedings that includes this cavefish paper. All right, here's a couple more questions that came in. What strategies can be used to get TOBD, or the theory of biological design, into the scientific community? Is there any strategies you suggest that we can use to get this into the general biological community, including probably the conventional biology community?
Well, one of the reasons why it's not really conventional is because people haven't been thinking on this. Our predominant emphasis has been to find problems with evolutionary thinking. And there's many of them. There's no doubt about it. But negative evidence alone will not dislodge evolutionary thinking for multiple reasons. I even wrote a paper about that came out in March of Acts and Facts on that. So we haven't been thinking in terms of theory of biological design. So the first step, the strategy is to get enough people thinking in terms of a theory of biological design. And somebody has to kind of like put something up on the board, get something up there, which is what we're trying to do at ICRs, get something on the board. And once you get a framework on the board, then it can be manipulated and tweaked and modified and made better and better and better. So it's to get it out there, get it on the board, get the discussion going, get some peer-reviewed papers in there. And another thing which would really help get it going is findings like we've had with our cavefish. Is actually get those findings and publish them out there. When people see a theory that makes sense, makes predictions, and it actually works, then I think the enthusiasm for it will build. Another strategy is to break down the barriers between engineering and biology. Now, biology is fundamentally and always has been reverse engineering. Don't take down those phony barriers that biologists do basic research and engineers do application. The reality is both are doing them both, and they're both melding together, and start to see biology as a gift to engineering, as a way to actually start analyzing creatures in terms of engineering. Let engineered man-made things that are doing similar functions guide your basic research into biology, help you find predictions of what you're finding. And when that turns out to be a useful research tool, it will also gain credibility as well.
And as you know, working at ICR, you've directed some of our staff, Dr. Jeff Tompkins and others, to work on a book, basically laying out all the non-random adaptations that are taking place. It's not random at all. There's more and more findings, as you presented tonight, of these non-random mutations that are really the rule. All right, let's move on. Oh, go ahead.
Okay, let me just close on that real quick. Great point. And that's change your thinking. Even creationists think in terms of random change, and random changes probably do happen, but it probably leads to broken systems. But when we're seeing purposeful genetic changes, we should start looking for directed genetic change and the mechanisms which may lead to that.
Okay, we've got one more question that just came in. Actually, maybe I'd do a couple more. Here's one that just came in. Do you think that different members of a biological population normally display differing levels of reproductive fitness? This kind of gets to the natural selection thing a little bit here. Do you think that different members of a biological population, can you see that at the bottom?
Yeah, I can read the question very well.
Okay.
Well, reproductive fitness, it'd be nice if we could determine what reproductive fitness is. In fact, fitness has been claimed to be an undefinable term in most dictionaries written by evolutionists themselves. But if you would, of course, within a population, there are different traits that exist in the members of those populations, which can solve environmental challenges differently. Some will be highly successful, some will be less successful, and some will be non-successful. Obviously, the ones with those traits that solve those problems will go on to live and go on to reproduce, and those traits will be passed on in the population of that. But what we really need to explain is, how do those traits solve those problems? And look for the genetics or the epigenetics behind those traits. So we're looking for the challenge, we're looking for the traits that solve those challenges, and we're looking for the mechanism that enables those traits. So what we really need to explain is, why is there differential reproduction, as some people might call it? Not that differential reproduction itself is a cause, but what the real thing that we need to explain is, why do we have differential reproduction? And in order to explain why we would have differential reproduction, you need to go back and look at the organisms. It's what's in the organisms themselves, which will either successfully solve problems or not.
All right, thank you. Another question just came in, and I have another one at least to go, but are you planning to write a book on this? And we talked about the non-random book, but is there going to be an all-encompassing Dr. Guliuzza's opus book that's going to put it all together? similar to what the other books that we put out at ICR.
Yeah. Yes. As much as possible, we will try to write a book on this because it needs to be written. More research needs to be done, more needs to be put in place, but we have a lot of that material already put together to put together a book. And what I really need is a little more time, in addition to the managing the organization as president, to find some time to actually compile all the material that's already been put together and put together a book. And the book itself would probably be in collaboration with other organizations which are working on a theory of biological design as well, because they have useful and thoughts to contribute to this idea.
All right, thank you. All right, here's kind of one last question. I think we're maybe running short on time, but this one kind of is maybe a lot of young Earth creationists have seen there's some controversy on this, so I'm going to put it out there. This was asked, can someone explain the difference between CET and the creationist standard view of natural selection, like you talked about? But how ICR originally on their displays in California had more of the traditional view. But can you explain the difference in kind of a short summary between CET and the creationist standard view of natural selection?
Sure. And I went through that because I wanted to answer this question actually in advance with ICR's original explanation on there. So we find that these fish have eyes, and now they no longer have eyes and pigmentation. And the standard explanation is that Darwin's mechanism, the selectionist mechanism, is actually true, and it works, but it can't do everything that Darwin claimed. In other words, that it is through random mutations, through copying errors within the population of the fish, which then gets sorted out through struggles to survive and a hit and miss process. That would lead to a trait which they would consider beneficial in the cave condition. And by the way, our standard explanation, which is given there, which is tossed in on many of these, is that organisms would maybe get diseases of the eyes, or they would crash into things with their eyes. That doesn't happen. That is the typical silly story which gets invented with Darwinian thinking in many ways to accommodate these things. But I've watched these cavefish now for years, and they never crash into things. They never bump into things. They don't scratch up their eyes on that. So this just doesn't even happen as well. So you actually have random mutations sorted out in a hit and miss process, being driven by the cave conditions, which would be called selective pressures, which eventually drives these fish to a condition of being blindness over a period of time. In other words, passive creatures being driven along by nature. CET, exact opposite. It doesn't see passive creatures, it sees active problem-solving creatures that have innate built-in capacity to detect problems, solve those problems, and when they successfully solve those problems, they fill different niches and they fill different areas. And what you need to do, if you need to critique this, is address the quotations and the information that is brought up by the evolutionists themselves and this particular model. But I think as we've seen tonight, it can actually make predictions, and these predictions can be verified. And it's a very different model. It's organism focused, not design and not environment focused. It's engineering based, fully engineering based, and not a trial and error, hit and miss process. So there's radical differences, which I think if you listen to tonight's talk, you could see there's some major ones.
Everything you said about the fish is true. One last thing, I think there's a question about, do you expect the loss of the eyes to be maybe reversible also?
Perhaps. Great question. First of all, we have to determine what is the mechanism that leads to the loss of eyes. We know it doesn't take millions of years or even hundreds of thousands of years, because they're not going to be able to live in the cave. And besides, it's not just a loss of eyes and not just a loss of pigmentation. There's a change in the fish heart, change in their red blood cells, change in their metabolism, a change in their shape of their skull, change in their jaw, change in their feeding behavior, there's so many changes which are actually specific to these fish, not just the two that we usually focus on, pigmentation and eyes. And they're probably all the fish detect certain changes in the cave, which then when they detect those, and we don't know exactly what those conditions are yet, or what the combinations of those conditions, or the percentage of the combinations of those conditions, what they exactly are yet, when they detect them, then these changes start into play on that. And when we determine that, then it's possible we may be able to determine what causes the eyes to disappear and perhaps how they might reappear again. Maybe the Lord's mechanism is that they would come back through hybridization. In other words, a blind fish hybridizing with a sighted fish. That may be the mechanism that he has put into place. Or it may be a mechanism where they can be switched back on. I don't believe all the information is lost and it's broken, but how that is actually regulated in terms of the regulation of those genes and what the biological mechanisms are between exposure to an environmental condition and what modulates the regulation of those genes or even the epigenetic factors, nobody knows yet. And that needs to be delineated. And the reason why nobody knows is because nobody's really asking the right questions, looking for those elements between exposure and response, delineating all of those elements between exposure and response, and nailing those down. And that's why ICR has a big job ahead of them.
Well, I think we're pretty much running short on time. I'm going to probably call it quits right here. And I just want to thank you again, Dr. Guliuzza, for still a wonderful and clarifying presentation. And thank you for your question or answers to the questions as well. I just want to make an announcement as a reminder. We want to encourage anyone watching to check out the Logos Research Associates website. On the website, you can learn about the various research efforts of all our associates and ambassadors and find information about their publications. Please take a few moments, browse the website, and consider supporting one or more of the exciting creation research projects that are underway through Logos Research Associates. While you're on our website, please be sure you have subscribed to the Logos email list so you can receive notifications about other upcoming presentations. When you subscribe, you'll also receive the Creation Evolution Headlines newsletter. I want to let you know that a recording of this video will be made available on our website so you can share it with others who may not have been able to join live tonight. And I'm not sure what our next presentation will be. There's been some talk, but I'm not going to speculate, so we're just going to leave it at that. But we'll have another speaker next month. And I would now like to close out our session tonight with a short prayer. You should join me in praying. Dear Heavenly Father, just want to thank you for tonight's presentation, that it went well. It was able to broadcast well on this network. I just thank Dr. Guliuzza for his dedication and his clear and insightful presentation that he gave us tonight, and for all the Logos Research Associates, that we all bring glory to Jesus through our research. I pray that this video will reach others who view it on YouTube and other channels and bring glory to you, our Savior, Jesus Christ. I pray this in your name. Amen.
Amen. Thank you.
Thank all of you for joining us, and I'll see you in our next broadcast.